All right, viral encephalitis, super common in animals. These are usually systemic infections. There are a couple agents that target the brain specifically, but much more common to be part of a systemic syndrome. So a few mechanisms of injury, ways that viruses actually injure the brain. You could get direct cytopathic effect, so that'd be direct killing of the cells. Things like distemper herpes do that. The presentation is going to depend on which cell population is being hit. So if you get neurons, you'll get selective neuronal necrosis. If you get oligos, it'll be dysmyelinating or demyelinating. Distemper is a classic example of one that causes necrosis to oligos and results in demyelination. They can also, some viruses target the endothelium, so they have kind of a roundabout way of inducing encephalitis by causing a vasculitis and hypoxia, edema, and then secondary necrosis of cells. And then you can also get infection during development when those precursor cells are migrating through the brain, and that's when you get like those congenital malformations, the cavitating encephalopathies, the hydranencephaly, and the porencephaly. Finally, you get persistent or latent infections. So there's several big categories of viruses that do this, primarily pestiviruses, retroviruses, because they actually integrate into your genome. And then herpes undergoes latency and reactivation with stress. Pathology, viral encephalitis, unlike bacterial and fungal, typically causes no grossly observable lesions. So we rely a lot on microscopy and neuroanatomic localization and clinical signs to tell us that there is disease in the brain. Inflammation is non-suppurative, so lymphocytes, plasma cells, some macrophages, and then you get that perivascular cuffing of inflammatory cells that are coming from the circulation to respond, but they get impaired by the dense neuropil, so they just accumulate around the blood vessel. You also get a gliosis, primarily composed of microglia, so your resident tissue macrophages will proliferate just like they do in any tissue that's inflamed, so you get a microgliosis. And then these can actually form little nodules. Glial nodules are more typically seen in viral encephalitis than any other flavors of encephalitis, those little actual nodules of microglia. Meninges in viral encephalitis, involvement is less common and typically less severe compared to bacterial. All right, so there are a lot of viruses that hit the brain, either directly or indirectly. Again, the pathology of that, from your guys' perspective, is going to be pretty non-specific. It's going to be non-suppurative encephalitis, myelitis, primarily. A lot of these diseases you can differentiate based on what I call these bonus effects. So all these things in colors, I guess. This is actually my board study slide for viral encephalitis. And I focused on how to tell them apart by these other things they cause. So you can see certain classes of virus are much more likely to cause malformation. So these would be in utero infections typically. There are certain, especially herpes, that tend to cause vasculitis and deal damage that way. There's quite a large number of demyelinating diseases, and notice that demyelinating and hypomyelinating diseases are also the ones that tend to cause malformations in a lot of cases. So it's kind of based on what's the whole picture in the whole animal, are there congenital signs? Am I seeing, vascular disease in other organs? I'm not going to go into this too much because you guys have the information, but this is a must-know. Canine Distempervirus, similar pattern for other paromyxoviruses of animals. So I've listed a couple syndromes down here that you hopefully will get to see on the floor. We do have a lot of seals and sea lions and dolphins and such. She's like, I've seen it. What's up? Yeah. Hypo is not enough. So hypomyelination is not enough myelin formed or it's abnormal. So that's a development thing, versus demyelination is it was there and then something happened and then we lost it. Yeah, ferrets, wildlife is very susceptible. So we also get a lot of wildlife here. Raccoons, skunkses. That's a skunk fox hybrid, in case you're wondering. Super common, always should be in the front of your mind if you're dealing with wildlife. Could it be canine distempervirus? And you can see here, same signs, crusty eyes. These guys could be brothers, right? His nose isn't quite as drippy. Yep. So most common syndrome that you guys are going to see is this pneumonia plus encephalitis, and that's kind of generally true in multiple species. That's a paramyxovirus, morbillivirus thing. Also demyelinating, also immunosuppressive because they cause necrosis of lymphocytes. So this is a multisystemic disease typically. This syndrome you're typically going to see in young, unvaccinated or under-vaccinated puppies because they're not old enough to have their full series. So just make sure you rule out seizures from hypoglycemia are also quite common in this age group. Look for parasites, another big cause of hypoglycemia and potential seizure activity. Yeah. Does demyelination cause neurons to die? Vote yes. Vote no. No, definitely not. That's primary demyelination. You lose your myelin, axon struggles a little bit, but it's alive. It struggles to conduct. It slows conduction because what's the function of myelin? It speeds up your conduction, right? So conduction will be slowed. You'll get neuroscience, but the axon is okay. The neuron's okay. Secondary demyelination, you start with death of the neuron or loss of the axon. And when the axon's gone, myelin disappears immediately because there's no support. So it depends if it's primary or secondary. All right, so there are a couple pretty uncommon. You're probably not gonna see these out in practice, but there are some other syndromes that distemper can cause. There's a post-vaccinal distemper encephalitis that we really don't see anymore. It's back when we used modified live strains. So if animals are infected as adults and are either unusually susceptible or unvaccinated, undervaccinated, they can develop this syndrome of multifocal encephalitis, and it's unusual because it's an adult. They don't typically present with the respiratory signs. It's just brain, so it's a little weird. But again, this is pretty rare. And then old dog encephalitis. So this is if you get infected with a replication deficient strain, so a CDV strain that's essentially sick and struggling and can't cause a fulminant infection and kill you like it wants to. It just kind of struggles along in the brain, and you get what's called old dog encephalitis. So they present with encephalitis, but it takes the whole life of the dog for them to develop enough disease to show signs. Again, super uncommon. Probably not going to see it, but should probably know about it for boards. All right, so we have distemper in a wolf. Here's a normal brain for reference. What is the issue or issues here? Dilated ventricles, so what do we call that? Hydrocephalus, do you think it's congenital or acquired? Why? Because he has a disease, Dr. Eames, dummy. Sure, yep. All right, what else? What's the other lesion? There's for sure loss of white matter. Look how thick this is, especially right here. This is internal capsule right here. Look how thin and sad this white. Look at his corpus callosum. It's like, here's your corpus callosum over here. So yeah, we've lost a lot of white matter. So lateral ventricles are moderately enlarged. The cortical white matter is diffusely reduced in thickness. That would be my descriptor. So how do you interpret these changes? We said hydrocephalus, and then what are some ways to lose white matter? Demyelination, pressure atrophy, hypomyelination, right? Maybe it was never there. Probably not, because I just told you he has distemper. So yeah, we've lost white matter. We've got hydrocephalus. This is a special hydrocephalus. We do have demyelination. This is called hydrocephalus ex vacuo. The ventricles are enlarged not to any obstruction, but because you've lost white matter, and that's made the ventricles big, and the space has just filled up with CSF because it's there. CSF is not causing any problems here. Got it? Cool. Hydrocephalus ex vacuo, not obstructive. All right, real quick, and then we're done. Parasitic encephalitis and myelitis. I break these into a couple different classes. So there's things that are there on purpose. They're in their either definitive or intermediate host. They want to be there. So they tend to not cause a lot of clinical disease in their natural host. These guys, they are not in their natural host anymore. These are aberrant or dead end hosts. And they are totally lost. They're trying to get somewhere that doesn't exist because they're in the wrong host. Does everybody know what this is? This is a human brain. See all the little lesions? Tapeworms, cook your meat, or become a vegetarian. These are all tapeworm cysts in this person's brain. Ew. Okay, so cysticercosis is what we call this syndrome. I want to spend a minute on Encephalitozoon cuniculi because I don't feel like you guys get enough rabbit stuff. And this is a classic rabbit Navli thing. So this is an obligate intracellular microsporidian. It is a fungus that acts like a protozoan. So it's kind of in that weird space between organisms. This is zoonotic and infects a broad range of species. Disease is most common in the rabbit because they are the definitive host. I've heard estimates that up to 90% of more rabbits are subclinically infected with this. So if you see a rabbit, you will see this disease for sure. However, since the rabbit is the definitive host, they're also they have a lot of subclinical infections, but that seroprevalence is high. So we know that most rabbits have been exposed or infected at some time in their life. This is the 90% or greater number of the seroprevalence. However, clinical infections are also common, often in immunosuppressed rabbits or younger rabbits or animals with other comorbidities or disease. These guys present with primarily CNS and ocular signs. So head tilt, rolling. Does anybody know what this posture is called right here? Starts with a T. Kind of twisty. Torticollis means your neck is twisted. So opisthotonos is back like this. Torticollis is like twisty. Yeah. This is almost an opisthotonos, but this one's a torticollis. I think they're kind of similar. And then this organism also infects the lens epithelium. So it causes what's called a phacoclastic uveitis. We talked about this a little bit in lab yesterday. Clastic, like an osteoclast, that means to break apart. And phaco is lens. So since lens protein is sequestered from the immune system, your immune system has not seen lens protein. If it gets a glimpse of your lens protein, look out, it will eat your lens. So that's phacoclastic uveitis. Trauma to the lens or injury to the lens causes exposure of lens protein. Immune system goes crazy. Lens melts. Eye gets inflamed. So initially they'll present with a cataract. Once the infection is severe enough, it'll actually rupture the lens. So here's you can see uveitis. Organisms are shed in the urine because lesions are primarily in the kidney. So this is the most common place to see gross lesions is in the kidney. Major differential for this presentation, this neuropresentation, the head tilt is Pasteurella mutacida, which is called snuffles in rabbits. It's another rabbit Navli disease for you guys. Any abscess anywhere in a rabbit, Pasteurella mutacida, number 1, 2, and 3 differential. So that results in very commonly otitis media interna that can present similarly with the head tilt and the rolling. So pretty much E. cuniculi and P. molt should be on your rabbit differential list for neurosigns. When we do see lesions in rabbits, they're most common in the kidney. So this on the left is an example of a more acute presentation, whereas over here you have a more chronic. This animal was probably subclinical up to death. Maybe it had some elevated renal values, but they can stay subclinical for their lifetimes. Acute presentation, you're going to get necrosis, hemorrhage. There's a little bit of pitting here. See how these lesions are kind of sunken down in, but all this red is hemorrhage and necrosis and tissue loss. In a more chronically infected case, again, you'll see pitting, but it's primarily due to fibrosis. The infection is much less active. It's just kind of smoldering along, causing slow damage. And all this streaking here is due to there being an interstitial nephritis. So there's lots of inflammation in there, fibrosis. There's another rabbit with a head tilt. Diseased rabbits are so cute. Quizzical. And then here's what those organisms look like in the tissue. So they actually will form these little intracellular cysts almost, and then the cell will eventually burst and release organisms. So that's where you're getting necrosis of cells. It's from lysis and release of bugs, preferably into the urine. That's where it wants to go. Okay, real quick, Neospora, Sarcocystis, grouped together because they're very similar. You can just kind of copy paste a lot of this. They're all apicomplexans. They rely primarily on these predator-prey host cycles. So example, carnivore to ruminant is a common one. Opossum to like small woodland creature. Cats and rats was the classic one for Toxoplasma. So usually the carnivore is the definitive host, and whatever it eats is the intermediate host. And if you develop disease, you're either the intermediate host or you're an aberrant host, probably. So, pathology, common pathology of protozoal disease. Protozoa typically result in tissue necrosis. So they will actually insist in tissues. This form doesn't cause any inflammation because the body can't really see it. It's sequestered. If these rupture and release, or if these organisms are like free swimming around and replicating, that's when you get tissue necrosis. So here's what toxo looks like. Here's a cyst in the brain. Looks fairly similar to encephalitozoan cyst, right? And this cyst is full of little organisms. So all these little dots in here, they're actually kind of bananas. Toxo is kind of banana shaped. These are all the tachyzoites. And once these are released, then you'll get inflammation. But right now, everything around here is pretty quiet. There's no inflammatory cells because they don't see this yet. Toxo also causes abortions. So here's necrosis of the cotyledons in a ruminant placenta. It's classic pepperoni placenta is the Toxoplasma lesion. It looks just like it. So that's the dots are the little multifocal necrosis. Pepperonis. Meningoencephalitis. So again, protozoal encephalitis may not cause a lot of grossly observable lesions, but this guy has a little bit of hemorrhage in his meninges. This is a primate, I believe. And here's a necrotizing pneumonia in a cat. So all these little depressed white foci are foci of necrosis. EPM, again, similar concept as Toxo Neospora. Horses are actually aberrant or dead-end hosts. So this bug is totally lost. It has no idea where it's going. We see spinal lesions more often than we see brain lesions, but both are possible. So presentation in infected horses is usually relatable to spinal signs. So paresis, muscle wasting. Here's a horse butt, and you can see very asymmetrical muscle wasting due to denervation. So he's losing nerve tissue, he's losing innervation in the muscles, and then his muscles atrophy because they don't have any stimulus. Here's an ataxic horse, and here's the lesion in the spinal cord. So hopefully everyone can see this discolored kind of red to pink. There's increased little blood vessels in here. This is malacia. It's now grossly observable softening. This is a focus of malacia. All right, Meningeal worm. I'm actually going to quit and not read through these last two because they're just, the information is there. You guys can go over it. Discospondylitis, last thing real quick. I know I'm out of time, sorry. So this is inflammation of the disc. It's in the name, discospondylitis. Plus or minus inflammation of the adjacent ends of these vertebral bodies, but it's primarily centered on the disc. Otherwise, we'd call it an osteomyelitis, right? So discospondylitis often accompanies osteomyelitis, inflammation of the bone, but they are different. If I have a vertebral abscess, it's osteomyelitis, it's not disco, unless it's on the disc. So this can happen in any species. It's most common in dogs, especially large breeds. And this is, the association of this is it's usually hematogenous bacterial infections, can be fungal, usually bacterial. These two underlying pathogens are the classics associated with lesions for some reason. They like to specifically go to the vertebra. I mentioned brucella likes to go to the lumbar vertebra in particular. You can also get these caused by trauma if you get direct inoculation of that space. Large ruminants with severe pleuropneumonia can get inflammation up actually into the vertebral bodies and disc spaces. On rads, you'll see collapse of the disc space, and then you'll see exudate and potentially spinal compression, osteomyelitis at the end plates. That's what it looks like. Normal discs, janky, angry, slightly widened or collapsed, irregular disc spaces, and kind of this moth-eaten lysis of the end plates. Pus in the disc space, so cool. This disc space is totally collapsed. This looks like the radiograph here, where you've got kind of the moth-eaten ends, and the exudate from the disc has actually squirted out and gone into the canal. And we made it.